Hello everyone, my name is Raj Mukhopadhyay. I'm a third year medical science student at Western University. Today I'm going to discuss with you the mechanism of infection of the virus that causes COVID-19, which is SARS-CoV-2. I'm also going to discuss with you the mechanism of action of a new drug that is being used in a hospital setting. It's called Remdesivir. So let's get started. So this virus that is shown here, this circular virus, this is the SARS-CoV-2, and this is the coronavirus. So this has these crown-like projections. These are spikes, and what these do is they allow the virus to bind to a cell surface receptor that's highly expressed in human epithelial cells, especially in the lung. This is why SARS-CoV-2 is a respiratory virus, because it really attacks the lungs. So these receptors are present in the lungs, and so this virus has affinity for this receptor, and it binds to this receptor, and then the virus gets into the cell via endocytosis. And then once it's in the cell, it deposits this RNA, and coronavirus such as SARS-CoV-2, it's a plus sense RNA virus. I will explain what that means in a second. So basically what that means is this yellow strand that you see here, once that is inside the cell, this strand is going to be used directly by the host ribosome. This is the host ribosome. So this is our ribosome, which is in our cells. Our host ribosome is actually going to translate this RNA from the coronavirus. So, and this initially will produce one viral protein, which is a protein which is called RNA-dependent RNA polymerase. RNA-dependent RNA polymerase. And this is viral, which means that it is not endogenous, so it is not in our human cells, because we never make RNA from an RNA. So this is what this protein does. So this protein will take this strand, and it'll make more copies of the viral RNA. So it's going to take this, and it's just going to make more. It's going to synthesize more. So that is what this does. That is the function of RNA-dependent RNA polymerase. It creates viral RNA. More viral RNA. Then all of this is going to be packaged, and it's going to be ready to infect more cells. And also these will code for more viral proteins, such as your spike protein and your head protein. And all of these will be translated by, again, our host ribosome. So, and once these will really cause the infection and really give you the symptoms. So, yeah, and once that is packaged and it leaves the cells, it's going to infect even more cells. So that is the basic description of how SARS-CoV-2 causes infection in humans. So now let's get into a new drug that is being studied. It is being used in very intensive hospital settings where patients are in critical condition. And it's actually been showing, some studies have been showing that it is producing a good effect. patients are having good outcomes from this drug, but I believe that more studies do need to be done for conclusive evidence. But so far, signs look good. So what is this drug? This drug is Remdesivir, right here. So this is the drug. And this, what type of drug is it? It's a prodrug, and it's a nucleoside analog. Nucleoside analog. So basically what this drug is all of our cells use nucleotides for replication. You may know that. So we have adenine, guanine, cytosine, thymine. This is in DNA. In RNA, we have everything is the same except uracil. Uracil. For RNA, we have uracil. But now, what remdesivir is, remdesivir actually mimics this one. This is adenosine. So this is ATP. And remdesivir actually looks a lot like ATP. You can see right here, structurally, remdesivir has it has this whole nitrogenous base. It has the phosphate. So it looks pretty similar to ATP, right, at a glance. But there's obviously some differences in the structure that you can see. This nitrogen is different. All of this stuff here is different. This group is different. So there's obviously differences in structure, and those are very important for the drug. So let me explain what remdesivir does. So remdesivir, it's given IV, so intravenous in the hospital. And once remdesivir gets inside the cell, it's inside the cell, and it's a prodrug. So this molecule right here has no activity. It gets metabolized into the active form, which does have activity in the cell. So this is the GS441524. That's the active form of remdesivir. This is what's going to give you the therapeutic effect of the drug. So yeah, once again, this looks similar to ATP, but there are differences, and these differences are exploited by us. So ATP is normally used by this guy, RNA-dependent RNA polymerase, to make new viral RNA. Now, when remdesivir is floating around in the cell, the active form, this RDRP is gonna look at this and think, hey, this is an adenosine. So let's put this into the growing strand. So let's imagine that RDRP is working and replicating this strand. Let's imagine it's making this strand, and as it's going, but it then incorporates remdesivir right here. This is remdesivir. Instead of an adenine, it puts remdesivir. Now, once it puts remdesivir, this will stop the viral replication. This stops the polymerase from continuing to replicate the strand, and then you don't get more viral RNA being produced. And then it obviously, binding is not perfect. I mean, it's obviously still gonna get the correct ATP sometimes. It's obviously not gonna get remdesivir every single time. So that is one of the considerations. I mean, it's going to slow the infection, but obviously the effect will depend on the dose and the concentration of this that is competing with ATP for the active site of RDRP. And another concern with this drug, but I'll explain why it's not really a concern, is that okay, we have many polymerases in our cell, normally used for transcribing RNA from DNA and such. So what if remdesivir goes and blocks a good polymerase that we need? So this may happen sometimes actually. Remdesivir, its unique structural elements allow it to bind better to RDRP than it would bind to a good polymerase that we don't want it to interfere with. So RDRP has a binding site. It binds the nucleotide, which is remdesivir in this case. So remdesivir has to possess some unique structural elements that will make RDRP bind better to remdesivir than it would to ATP, or also remdesivir, these structural features in remdesivir are basically kind of matching or similar to, or complementary I should say, to allow for better binding to the binding site of RDRP itself. So this binding site in RDRP probably has unique features, and these features on remdesivir make it a good match, and that will not be the case for our good polymerases, which we don't want remdesivir to bind to. So that is one of the ways we can ensure that we're not messing up our replication with our normal polymerases that we use in our cells, and we are really targeting this bad guy, which is the RNA-dependent RNA polymerase.